Hi everyone, today I would like to teach you about a special field in science and some of the terms used in it.
Functional genomics is a field of biology that aims to understand the function and regulation of genes and their products on a genome-wide scale. Here, I would like to introduce to you 25 frequently used terms in functional genomics. Genome is the entire genetic material of an organism, including all of its genes and non-coding sequences.
Transcriptome is the complete set of RNA transcripts produced by the cells of an organism representing gene expression profiles. Proteome is the complete set of proteins expressed by an organism, including their post-translational modifications. Metabolome is the complete set of small molecule chemicals found within a biomolecule sample. DNA sequencing is the process of determining the order of nucleotides in a DNA molecule. Gene expression is the process by which information from a gene's DNA sequence is used to produce a functional product such as a protein or RNA molecule. Promoter is a region of DNA that initiates the transcription of a specific gene, typically by binding transcription factors. Transcription factor is a protein that binds to DNA and regulates gene expression by either activating or repressing transcription. Chip sequence is a chromatin immunoprecipitation followed by sequencing used to identify DNA sequence associated with specific proteins, such as transcription factors. RNA interference is a mechanism by which small molecules of RNA inhibit the expression of specific genes. Single nucleotide polymorphism is a genetic variation where a specific DNA base differs between members of the same species. Genomic marker is a DNA sequence with a known physical location on a chromosome used to identify individuals. DNA marker is a mutation in a SNP or variable number of repeats of base pairs that are landmarks in the genome. Microarray is a high-throughput technique that allows simultaneous measurement of the expression levels of many genes in a single experiment. Sanger sequencing is a technique of DNA sequencing in a segment of DNA based upon selective incorporation of chain-terminating deoxynucleotides. Next-generation sequencing is a massive parallel sequencing method used to determine a portion of the nucleotide sequence of an individual's genome. CRISPR-Cas9 is a revolutionary gene editing technology that allows precise modification of DNA sequences in the genome. CRISPR-screen is a technique that uses CRISPR-Cas9 to systematically target and perturb genes in order to study their functions. Functional annotation is the process of assigning biological meaning or function to genes or their products. Functional screening is a method used to identify genes or proteins involved in specific biological processes through systematic experimentation. Gene ontology is a standardized system for categorizing genes based on their molecular function, biological process, and cellular components. Gene knockout is the complete removal or inactivation of a specific gene to study the effects on an organism or cell. Pathway analysis is the study of how genes and their products interact within biological pathways to carry out specific functions. Epigenetics is the study of hereditary changes in gene functions that do not involve changes to the DNA sequence itself. Functional Genomic Database is a repository of genetic data and annotations often used for data mining and analysis in functional genomic research. These terms are fundamental in the study of functional genomics and provide a foundation for understanding how genes and their products function within living organisms. That is all. Thank you for watching, and remember, there is always something new to learn. For more information, you can search for it in the links in the description.